Hi, everyone. We are waiting few seconds to give the opportunity to all the people to connect to the webinar. Okay, so welcome everyone to this trainee talk series. Today, as usual, we have selected 3 speakers, Pauline, Parham, and Jan. And just a couple of advertisements. So please save the date of our next Cancer Cachexia conference that will be held in Torino, Italy, at the end of September 2025. And also, I would like to remind you our next seminar in October 9. The speaker will be Mauricio Berrier-Diaz. And Mauricio will show data revealing a new mechanism by which the liver contributes to tissue loss in cancer cachexia. So I remind you that you can put your question in the Q&A box. I will read your question and the speaker will answer your question in live. So let's start with the first speaker, who is Pauline Morigny. So Pauline, please, you can share your screen, your presentation.
Yes.
Perfect. So just a couple of words to introduce Pauline, that did a PhD in the field of obesity and type 2 diabetes in France. And in 2018, she joined the Institute of Diabetes and Cancer of Helmholtz, Munich, to work on cancer-associated cachexia. During her first postdoctoral experience with Mauricio Berrier-Diaz, she worked on the identification of tumor-secreted factors as hallmarks of cancer cachexia. And since 2021, she has been studying the involvement of bioactive lipids in cancer cachexia pathogenesis in the group of Maria Rom. So, Pauline, you have 15 minutes, and please, the virtual stage is yours.
So good evening from my side. Thank you, Roberta, for the nice introduction. And I would also like to thank the Cancer Cachexia Society for this great opportunity of presenting my work today, which indeed focus on the contribution of bioactive lipids to cancer cachexia. So in Maria Rom's lab at Helmholtz Munich, we are particularly interested in the study of tissue metabolism and crosstalks in cancer cachexia, and we aim at identifying new pathways and mediators that could contribute to the pathogenesis of this syndrome. So when we started this project a few years ago, we already knew several tumor and host-secreted proteins that could contribute to cachexia, meaning contribute to the loss, for instance, of fat and skeletal muscle mass. However, not so much was known about alteration in lipid composition and especially in bioactive lipids in cachexia, although bioactive lipids are really important signaling molecules. So this is why a few years ago, together with my former colleague, Julia Geppert, we had performed a broad range lipidomic analysis of different mouse models of cancer cachexia, as well as from cachectic and weight-stable patients with cancer. And back then, we had submitted plasma samples to our lipidomics core facility. And thanks to the lipidizer platform, we were able to study 30 different lipid classes and over 1,100 different lipid species. From this analysis, we identified high circulating levels of sphingolipids and especially of different ceramides and modified ceramide species as a hallmark of cachexia, both in mouse models, but also in cachectic patients with cancer. And we have also observed that ceramides were already upregulated at an early stage during cachexia development, and that they positively correlated with the extent of body weight loss in cancer patients. Back then, also, we were interested to know which tissue might contribute to this increase in circulating ceramides, and this is why we had performed some qPCR analysis to assess gene expression of ceramide synthesis enzyme in different metabolic tissues and tumors. And we observed that, indeed, there was an increase in the expression of ceramide synthesis enzyme in the livers of cachectic mice here, and compared to either healthy PBS-injected mice or non-cachectic NC26 tumor-bearing mice. And interestingly, this increase in ceramide synthesis was only observed in liver, since we observed rather a decrease in adipose tissue or in skeletal muscles. So overall, from this study, we had identified increased circulating ceramides levels as a hallmark of cancer cachexia, both in mice and in patients. And increased ceramides levels very likely arise from increased synthesis by the liver. These results were of particular interest for us because ceramides had already been associated with a lot of pathophysiological processes in other diseases. Indeed, they had been reported to promote mitochondrial dysfunction, oxidative stress, and liver steatosis. They had been associated with the development of insulin resistance and ER stress in adipose tissue and skeletal muscle, as well as to sarcopenia. They had also been associated with apoptosis in heart. They also promote systemic inflammation and altered feeding behaviors. And since all those effects are also a hallmarks of cachexia, we therefore wondered whether increased ceramide synthesis in cachexia could contribute to its pathogenesis. And this is the goal of my actual project. So therefore, first of all, we wanted to know whether our hypothesis was relevant for cachectic patients with cancer. And thanks to a collaboration with Hans Ohner from Klinikum Recht der Isar in Munich, we had the opportunity to get liver, adipose tissue, and skeletal muscle samples from patients with cancer. And so these patients, as you can see here, suffered from gastrointestinal cancers and with different degrees of cachexia. So the patients were stratified based on their cachectic phenotype, so either non-cachectic based on Firon's definitions of cachexia, or either suffering of mild cachexia or strong cachexia when body weight loss was above 10%, as you can see here with the body weight loss in the past 6 months. And with this tissue, we performed some qPCR analysis of ceramide synthesis enzymes, as well as liver lipidomics. From those samples, we observed a strong induction of ceramide synthesis enzyme expression in the liver, which was positively associated with the extent of body weight loss in patients. And this is what you can also see here on this correlation map. So here you can see the correlation coefficient, and you can see that expression of ceramide synthesis enzymes in the liver was positively associated with the extent of body weight loss in the patients. Interestingly, induction of ceramide synthesis enzyme was not visible in other tissues, such as visceral, subcutaneous adipose tissues, as well as in skeletal muscles, so same as in our preclinical model. And accordingly, we did not observe such a strong association with the extent of body weight loss in those tissues. So from those results, it really seems that also in patients, ceramide synthesis is increased specifically in the liver. And to further strengthen these observations, we also performed liver lipidomics from the same patients. And here also we observe increased total levels of ceramides, as well as increased level of specific ceramide species. Accordingly, hepatic levels of these ceramide species were positively associated with the extent of body weight loss in these patients, but also with the expression of ceramide synthesis enzyme in the liver, confirming the association between cachexia and increased liver synthesis of ceramides. So then afterwards, we wondered whether ceramides could then contribute to the development of cancer cachexia. And to answer that question, we decided to perform an in vivo experiment in the well-established C26 mouse models of cancer cachexia. So to this end, mice were injected either with PBS or C26 cancer cells and were treated either with a vehicle or with a ceramide inhibitory treatment, which is Myriocin. And then we we prepped the mice at an early stage during cachexia development, so as soon as they reached more than 10% of body weight loss. As you can see here, our treatment was really efficient to counteract ceramide synthesis in C26 tumor-bearing mice. Treatment did not affect tumor growth nor food intake. However, we observe a trend towards an improvement in body weight loss in these animals. This improvement in body weight loss was not explained by a change in fat mass, but rather by a change in lean mass, since we observed some improvements in heart and skeletal muscle weights. And accordingly, we also observed functional improvements in these mice, as you can see here with the grip strength at endpoint. So overall, inhibition of ceramide synthesis led to moderate improvement in cachexia at an early stage, and therefore we wonder if this beneficial effect would strengthen in more advanced cachexia. So to this end, we have performed in collaboration with Bert Blow from the Wim Institute of Padova another experiments with the same settings, but this time the mice were prepped at a more advanced cachexia stage, so as soon as they reached more than 20% of their initial body weight. Once again, the treatment was really efficient to counteract ceramide synthesis in C26 tumor-bearing mice. And once again, the treatment did not affect tumor growth. However, it strongly improved body weight loss in C26 tumor-bearing mice. Once again, this improvement in body weight loss was not explained by a change in fat mass, but rather by an improvement in lean mass with increase, for instance, heart weight and weights of different skeletal muscles. And this was again associated with some functional improvement as measured by grip strength. So then we wanted to dig more into mechanisms in order to understand how ceramides could lead to these beneficial effects. So first of all, we had a look at the muscle where we observed a very strong phenotype. And in the advanced cachexia experiment, we observe a very strong inhibition of atrogene induction by myosin in different types of skeletal muscles. And inhibition of atrogenes was also associated with a significant reduction in the total levels of ubiquitinated protein, suggesting that there is a lower protein degradation in the muscle. However, such an effect in advanced cachexia was mitigated when we looked at the early cachexia experiments. So in the end, this suggests that ceramides have those beneficial effects in advanced cachexia, but this is probably not the primary effect by which ceramides have this beneficial effect. What I mean here is that it is probably not the primary trigger. So therefore, we also had a look to insulin sensitivity in the muscle, but here also we did not observe any difference in insulin signaling in the muscle. So therefore, we believe that the beneficial effect that we see at an in our early cachexia stage, in our first experiments, probably comes from the primary tissue where ceramides are produced. So this is why we then had a look to the liver in the early cachexia experiments, and we started to check again the classical markers of cachexia. And here again, as you can see, we did not observe any effect of myosin on the expression of acute phase response protein. So therefore, we wondered if ceramides could interfere with insulin signaling and with the anabolic effect of signaling of insulin. However, here we also did not really observe any effect of ceramides on insulin signaling in the liver. And therefore, we just wonder if our C26 tumor-bearing mice were developing insulin resistance. And to answer that question, we have performed an insulin tolerance test in either PBS-injected mice or cachectic C26 tumor-bearing mice. And as you can see here, in the end, our C26 tumor-bearing mice are not really insulin resistant here. So this is probably not the way ceramides induce contributes to cachexia pathogenesis. So in the end, since ceramides do not seem to affect the classical markers of cachexia, especially at an early stage of the disease, we therefore dig into the literature and hypothesize that ceramides might interfere with mitochondrial function and energy production at the early stage of cachexia development. So to answer that question, we have performed a subset of in vitro experiments in which we treated primary hepatocytes and C2C12 myotube with a short ceramide analog, which can be easily incorporated into the cells. And we have performed a seahorse analysis to measure mitochondrial function. Here, we observed a dose-dependent decrease in mitochondrial respiration upon ceramides treatment, and this led to reduced ATP production into the cells. Also, ceramides induced the expression of some mitochondrial stress markers in both cell types, and in myotube, this contributes to myotube atrophy. So overall, it seems that ceramides in cachexia could contribute to atrophy and the cachectic phenotype by interfering with mitochondrial function. So finally, we wanted to bring a proof of concept in vivo that indeed ceramides could interfere with the mitochondrial phenotype. And to this end, in the early cachexia experiment in liver, we have performed in collaboration an electron microscopy analysis of mitochondrial morphology in C26 tumor-bearing mice treated or not with a ceramide synthesis inhibitor. Here, we have observed that in cachexia, there is an alteration in mitochondrial morphology. Mitochondria are less circular, and this was rescued, or let's say back to normal, when we treated the mice with myosin. To support these results, we have also performed a confocal microscopy analysis of mitochondria connectivity, which is also a way to assess mitochondrial function or estimate. And as you can see here, also in cachectic C26 tumor-bearing mice, mitochondria were less connected. And this was also rescued by myosin, suggesting a better mitochondrial function. In addition, we have also observed increased expression of mitochondrial stress markers in the liver of cachectic mice, which was partially rescued by myosin. And ultimately, in order to estimate mitochondrial function in vivo, we have performed a metabolomic analysis to measure the TCA metabolites in the liver, assuming that if the mitochondria is more active, we would have higher levels of TCA metabolites. And this is actually what you can see here. So indeed, it seems that ceramides can alter mitochondrial function in vivo in tumor-bearing mice at an early stage. So overall, in this project, we have identified a potential new mechanism contributing to cachexia. We have shown that early on, ceramides synthesis is increased in the liver and can interfere with mitochondrial function and energy production, and that in more advanced stage, ceramides can also contribute to the loss of lean mass and especially of skeletal and heart mass. And we believe that this effect might be maybe the consequence of increased circulating levels of ceramides and transfer between tissues, but also the consequence from altered whole body energy homeostasis by the liver. And with that, I would like to really thank my supervisor, Maria Rom, for offering me the opportunity to work on this great project. I would like also to thank all my colleagues and collaborators who are supporting us with this project. And thank you for your attention, and I would be very happy to take questions.
Thank you, Pauline, for the great presentation. So we have a question from Fabio Penna. So he is asking if are interleukin-6 circulating levels and or muscle transcripts affected by ceramide synthesis blockade? In other words, is the inflammatory status affected by your treatment?
So we have not dig too much into that direction. What I did in the experiment was to measure circulating IL-6 levels, and this was not affected by the treatment. But this is the only thing we have measured so far.
Okay, thank you. So we have another question from Avontik Bazak. I apologize for the pronunciation. So hi, Pauline, excellent work and nice presentation. Have you seen any changes in mitochondrial lipidomics, especially in cardiolipin species?
This is actually a very interesting question as well. This is ongoing. Actually, we are planning to do mitochondrial lipidomics. So we're going to know soon.
Okay, and may I ask you if you have any mechanistic hypothesis on how ceramides can have an impact on mitochondrial function, or if you are exploring some mechanistic hypothesis?
So it has been published a few years ago that ceramides can directly interact with some fission proteins, and that they induce mitochondrial fission. And therefore, this is how they can interfere in part with mitochondrial function.
Okay. So we have another question from Margaret Park. So really fantastic work. So does the mouse diet, so high-fat diet or standard diet, affect ceramides / cachexia in this model?
It's hard to answer that question, actually. It is known that high-fat diet can induce ceramides levels. However, it's hard for me to understand that, to answer that question, unfortunately. It could be that my hypothesis is that high-fat diet, by increasing even more ceramides levels, would aggravate cachexia or accelerate it, but we haven't assessed that.
Then going back to the human data, maybe I have missed this point. So you can see correlation between the level of ceramide synthesis enzymes in the liver with the body weight loss. And what about the muscularity? So the skeletal muscle index or the muscularity of the patients? I'm not sure if you show the data. If yes, sorry for the question.
No, and it's true that we haven't tried this correlation. It's actually really interesting. Yeah, we have the information whether the patients were sarcopenic or not. So this is something indeed that we can assess. But yeah, then I don't have the answer now.
Okay, and then going back always to the human data, if I remember correctly, you say an increase in ceramide circulating levels only in severe cachectic patients, right? I am wrong?
It's where it's the most pronounced, indeed.
Okay, so this seems to suggest that this could be a consequence, not the cause. Can you comment on that? So the consequence of cachexia and not something that is driving or that is implicating in the pathogenesis of the disease. Can you comment?
Indeed, I don't think it's a mechanism which is involved in the initiation of the disease. However, I think that once they are increased, they are clearly aggravated. And I think that's also what our in vivo experiment shows, that really the beneficial effect of inhibiting them is really in advanced cachexia. So I think, yeah, they are induced once body weight loss is initiated. This is what we see in animal models. But then I think we clearly show that it's able to counteract advanced cachexia.
Okay, it's clear. So thank you, Pauline.
Thank you.
Thank you very much for this very great presentation. So if you can stop, okay, sharing your screen, you can move with the second speaker, who is Parham Diba. Okay, perfect. So Parham, just a couple of words. So Parham Diba earned his bachelor's degree from the University of Oregon, and following graduation, he spent 3 years as a research assistant in Dr. Stephen Beck's lab at Oregon Health and Science University before entering the MD PhD program in 2019. His graduate thesis explores the effects of cancer and cancer cachexia on cardiac health. And the title of his presentation is Decoding Cardiac Immune Response in Pancreatic Cancer Cachexia. So Parham, please, the stage is yours.
Thank you for the introduction. I also want to thank the Education Committee for the invitation. So I'll be presenting a part of my dissertation project, which focuses on the role of immunity in cancer cachexia-induced cardiac wasting. So as you all know, cachexia is a multi-organ syndrome, and very similar to skeletal muscle, cardiac muscle also undergoes significant wasting in patients with cachexia. And this can be appreciated by looking at the graph on the left side of the screen. And in this graph, when you compare healthy controls, which is in blue, to patients with cancer cachexia, which is in yellow, you can see this significant loss of left ventricular mass. And in the same study, they also showed that this loss of left ventricular mass is associated with poor survival. And you can see that here, where patients with a lower left ventricular mass also had lower survival compared to patients with a higher left ventricular mass. But in addition to cardiac wasting, there are also multiple studies demonstrating subclinical myocardial damage in patients. And for today, I'm only summarizing a couple of these studies. So in a prospective study that was looking at treatment-naive patients with colorectal cancer that also did not have any prior history of cardiac disease, when they looked at the heart, they found a significant reduction in the left ventricular posterior wall thickness. And again, this is consistent with cardiac wasting. But they also looked at cardiac biomarkers, and they found a mild but significant elevation in creatine kinase MB and high sensitivity troponin T. And this indicates that there's also myocardial damage in patients. But when they looked at ejection fraction, they did not see any signs of systolic dysfunction. And the authors concluded that this elevation in cardiac biomarkers is probably due to presence of microlesions on the heart. In a separate prospective study that again was looking at treatment-naive patients, they found very similar results, but they also showed that this mild elevation in troponin is associated with poor survival. And you can see that here, where patients with this mild elevation in troponin also had lower survival compared to patients with relatively normal troponin levels. And so even though we know the heart is undergoing atrophy and damage in patients, we don't fully understand the mediators of this damage, and that's what my project or dissertation work focuses on. So to investigate this question, we're using a murine model of pancreatic cancer-associated cachexia, and to generate the model, we orthotopically implant either sterile saline or a pancreatic cancer cell line called KPC cells directly into the pancreas. And from here on, I'm going to refer to these groups as either sham animals or PDAC animals, which is short for pancreatic ductal adenocarcinoma. And you can see when we looked at our PDAC animals, both by direct tissue analysis and also by echocardiography, we found significant cardiac atrophy. When we looked at plasma troponin levels, we found a mild but very significant elevation in troponin. And finally, when we looked at ejection fraction, similar to human patients, we did not see any signs of systolic dysfunction. So everything we see here is very similar to what we see in human patients, and we thought this was a good model to use to investigate our questions. So to better understand the mediators of wasting and damage, we performed bulk RNA sequencing of the heart both at 10 and 14 days post-implantation. And by looking at this heat map, we can see that some of the highly upregulated pathways are associated with an inflammatory response. And since the heart is enriched with different types of immune cells, we decided to investigate the immune landscape of the heart in our tumor-bearing animals. So we performed flow cytometry in the heart, and we found infiltration of neutrophils into the heart. And as you can see, we performed a time course study, and we found that these neutrophils infiltrate the heart starting at 7 days post-implantation. And then we further characterized these neutrophils, and we found that they are positive for CD244, which is a marker for granulocytic myeloid-derived suppressor cells. And these cells are basically a less differentiated form of neutrophils that have immunosuppressive properties. So they're functionally a little bit different than fully mature neutrophils. And to put this in a little bit more perspective, when we looked at the hearts of our PDAC animals in our time course study, you can see that at 7 days post-implantation, that's when we started to see a drop in cardiac mass. And by 10 days post-implantation, this drop in cardiac mass was significant. And when we looked at troponin, we found that this biomarker was significantly elevated starting at 7 days post-implantation. So the timeline for our cardiac phenotype is aligning really well with when we see these neutrophils are infiltrating the heart. And one of the things that we wanted to know was whether these infiltrating neutrophils are regulating the damage and atrophy that we see. However, we had a confounding variable in our experiments, and that is food intake. So by looking at this graph, you can see that our PDAC animals also demonstrate a decrease in food intake, and that also starts to happen around 7 days post-implantation. So before investigating the role of neutrophils, first, we wanted to know if this decrease in food intake plays any role in cardiac wasting and damage that we observe in our animals. So to answer that question, we performed a pair feeding study where we had sham animals that were on an ad-lib diet, and we also had sham animals that were pair-fed to our PDAC animals. And then at the end of the study, we split the heart in half, and we used half of it for qPCR, and then half was used for flow cytometry. So when we looked at cardiac mass, we found that there was significant atrophy both in our PDAC animals and also in our sham pair-fed animals. But when we looked at genes that are involved in cardiac atrophy with qPCR, you can see that we found a higher upregulation of these genes in our PDAC animals compared to our sham pair-fed animals. So this data suggests that, yes, the decrease in food intake does participate and does play a role in cardiac atrophy. However, there are also food-independent factors that are also playing a role in cardiac atrophy in our PDAC animals. We also looked at troponin, and you can see that this biomarker was only elevated in our PDAC animals. And again, that this suggests that this elevation in troponin is completely food intake independent. And when we looked at neutrophils, you can see that these cells only infiltrated the hearts of our PDAC animals. So after we had this data, our next question was whether these infiltrating neutrophils are regulating the damage and atrophy that we see. And very interestingly, very recently, a paper was published in a lung cancer cachexia model. And in their model, they also observed infiltration of CD244 positive neutrophils into the heart. And when they depleted these neutrophils, they're able to reverse cardiac wasting. So this was in a lung cancer cachexia model, and now we're in pancreatic cancer, so completely different tumor type. And we wanted to see if we can see the same phenomenon when we deplete neutrophils. So then we started performing some neutrophil depletion studies, and we used a couple of different depletion strategies. So one of the first strategies that we used was utilizing depleting antibodies. And we performed a 2x2 study where we had sham and PDAC animals that received a cocktail of depleting antibodies. And we also had sham and PDAC animals that received IgG controls. And with this strategy, you can see that we're only able to achieve a partial depletion in our PDAC animals. And when we looked at the hearts of these animals, you can see that with a partial depletion, we are not able to reverse cardiac wasting. And similarly, when we looked at troponin stayed elevated in our partially depleted PDAC animals. But what was interesting and kind of unexpected, we also found a significant elevation in troponin in our sham depleted animals. And this suggests that the neutrophils that are present in a healthy heart may have a baseline cardioprotective role. So since this method did not give us a robust depletion, we followed this with another depletion strategy where we used the chemogenetic approach. And in this approach, we crossed homozygous ROSA 26 IDTR mice with animals that were hemizygous for MRP8 Cre. And as a result, we were able to express diphtheria toxin receptor on neutrophils. And again, we performed another 2x2 study where we had sham and PDAC animals that only received the ROSA 26 transgene and did not express diphtheria toxin receptor. And we also had sham and PDAC animals that received both of these transgenes and did express diphtheria toxin receptor. And all of the animals in this study, so all 4 groups, received daily injections of diphtheria toxin starting at 3 days post-implantation. And with this strategy, you can see that we're able to achieve a robust depletion both in our sham and PDAC animals, and we perform flow cytometry both in the heart and in the blood, and we're able to deplete the cells from both of these tissues. Then when we looked at our animals, one of the first things that we noticed is that our neutrophil depleted animals experienced significant loss of body mass throughout the course of the study. And when we looked at their cardiac mass, you can see that even with a full depletion, we are not able to reverse cardiac wasting. And when we looked at troponin, again, we are not able, you can see that troponin stayed elevated in our PDAC depleted animals. And very similar to our antibody depletion method, when we looked at our sham depleted animals, again, troponin was significantly elevated in this group. And again, this indicates that the neutrophils in a healthy heart may have a baseline cardioprotective role. We also looked at skeletal muscle and white adipose tissue, and you can see that neutrophil depletion also did not reverse tissue wasting in these animals. And I would say overall, we had more wasting in depleted animals compared to non-depleted animals. And finally, when we looked at tumor mass, you can see that our PDAC depleted animals had significantly smaller tumors. And this is interesting because even though we reduced tumor burden in these animals, we still were not able to reverse cardiac wasting and damage. So one of the final questions that we had, we were wondering if these cells are initiating cardiac disease rather than being involved in the progression of the disease. And if you remember from our time course study, we started to see our cardiac phenotype on day 7 post-implantation, and that's also when the neutrophils started infiltrating the heart. So to investigate if these cells are involved in the initiation of the disease, we performed another 2x2 study, but this time we euthanized the animals at 7 days post-implantation. And in this study, when we looked at the heart, you can see that in both PDAC groups, the hearts have started to waste, but it's still not significant. And when we looked at troponin levels, again, troponin is significantly elevated in both PDAC groups. So this data suggests that these cells are also not involved in the initiation of cardiac disease. So in conclusion, we showed that PDAC-induced cachexia leads to cardiac wasting and damage. And this is accompanied by infiltration of CD244 positive neutrophils into the heart. However, depletion of these cells was not able to reverse cardiac wasting and damage. And this is different than what has been reported previously in a lung cancer cachexia model. And we think that even though the same cell type is infiltrating the heart, depending on the type of tumor present, they may have different function. And we think in pancreatic cancer, these cells are going to the heart as a secondary response to a damage that has already occurred, rather than being the regulator of the damage and wasting that we see. And finally, we showed that the neutrophils in a healthy heart have a baseline cardioprotective role. So with that, I would like to thank everyone who has helped with this project, including everyone in the Grossberg lab, and also all the funding sources that made the project possible.
Thank you, Parham. Terrific talk.
Thank you.
So if you have questions, please put in the Q&A. So Parham, you say that you saw a decrease in the tumor mass, correct?
Yes.
And this is not sufficient to mitigate the cardiac damage or tissue wasting.
Yes.
Correct. So can you comment on that?
Yes. I mean, the reduction in tumor mass was small, but it was still significant. So I think that just indicates that the size of the tumor doesn't necessarily dictate the severity of cachexia.
Yes.
So there might be the factors that are initiating wasting might be induced independent of the size of the tumor. But I still don't know what those factors are.
And what about the level of inflammatory cytokines between the two animal groups? Did you find any differences?
Yeah, that's a good question. That's something we still have not looked into, but that would be a good thing to look into to see if there's any difference in inflammation.
Okay, so it could be a good point. So we have a few questions in the Q&A box. So Joanna Karazinska is asking, do you know if circulating neutrophils or neutrophils to lymphocyte ratio is associated with cardiac cachexia in PDAC patients?
In PDAC patients, I would say there's not much data on the heart and the association of circulating neutrophils to the heart. So as far as I know, that data doesn't exist in patients. And I would say an increased neutrophil to lymphocyte ratio overall is associated with poor outcomes. But again, I don't know if there's any data looking at that ratio and see if that correlates with the amount of cardiac wasting.
Okay. Then Maria Roma is asking, how specific is the neutrophil depletion to the heart? I think general increase in neutrophil numbers in different tissues has previously been shown. So how specific is the depletion of neutrophils to the heart?
Oh, to the heart? It's not specific. So it's depleting it everywhere in the body, including even some of the neutrophils in the bone marrow. The thing is that it is extremely difficult to deplete neutrophils in PDAC animals. And with the technology available today, we can just do a global depletion of neutrophils.
Okay.
Yeah, so it's very difficult to just deplete neutrophils in the heart, but that's a good point.
Yeah. So, okay, I had the same question. So do you have any data about macrophages?
Yeah, that's a good question. So we have started looking at other immune cell types to see if we see any changes in other cell types, but I don't have data on that today. I would say in the lung cancer cachexia model, when they looked at macrophages, they didn't see any changes in macrophages in the heart. But again, since we're seeing such drastic differences in our models, it might be different in our model. But yeah, I don't have a lot of data on that today.
Okay.
Yeah.
So Fabio Penna is writing, thanks for your great talk. So nice to know that neutrophils are infiltrating almost every tissue. So others reported in the liver, brain, skeletal muscle. Can I just ask you the age of the mice? So is cardiac mass reduction the result of wasting or impaired growth?
Yes, so we did our experiments on mice that were around 12 weeks of age. And I think by that time they are, as far as I know, please correct me if I'm wrong, they're fully adult. So I think it should not be a result of stunted growth.
Okay, so thank you, Parham. We have to close here the Q&A session. Thank you again for your great presentation. So we can move on with the last but not least speaker, Jan Soon from Maastricht University. Perfect. So Jan used to be a doctor before she started her PhD. And recently she completed her PhD thesis on body composition in cancer cachexia research. Her focus is on exploring body composition combined with clinical characteristics to predict clinical outcomes. Additionally, she also investigates tumor metabolic activity via PET-CT in this area. So, Jan, please.
Yes, can you hear me?
Yes, perfectly, and we can also see your presentation.
Yeah, hi everyone, I'm Yan. I'm a PhD student, supervise Stephen O. Damink and Sander Rensen. It's a great honor to be here. I'm excited to show our study focus on body composition and immunotherapy in cancer cachexia. Recent years, immunotherapy has been developed and achieved remarkable success. During tumor genesis, the tumor receptor can recognize and respond to tumor-associated antigen, and the immune checkpoint protein like PD-L1 and PD-1 play key roles. The PD-L1 expressed on tumor cells can bind to the PD-1 receptor on activated T cells, thereby inhibiting cytotoxic T cells. Based on this, then the tumor cells can escape the immune detection and clearance. Based on this pathway, the PD-1 and the PD-L1 immunotherapy can play a significant efficacy in various cancers, including lung cancer and gastric cancer. However, lung cancer and gastric cancer still face a big challenge due to the tumor cells heterogeneity, which impact on the development of cancer cachexia. So why is immunotherapy not always effective? The potential reason is the host phenotype of patients interact with immunotherapy. The immune-related adverse events may aggravate the disease process and increase the risk of cancer cachexia. In another hand, cancer cachexia can also affect the efficacy of treatments. So it is critical to understand how tumor and host factor interact and influence immunotherapy. We know the tumor drive pro-inflammation cytokines such as TNF-alpha, IL-6, and IL-1 beta. They will impact the different tissues, including adipose tissue and skeletal muscle, affect the catabolism. Previous studies show that the systemic inflammation can promote the skeletal muscle wasting and the adipose tissue remodeling. And the recently study shows that the cytokines and infiltrating immune cells can also contribute to adipose size and the myocytes, their differentiation and function, as well as a systemic metabolic balance. So we are urgently to find an easy, fast, cheaper way to really deeply understand cancer cachexia. As AI technique developed in radiology, the body composition based on the CT scan can really support us to understand cancer cachexia very well. Look at the CT scan after the analysis. The red color is the skeletal muscle, and the yellow color is the visceral adipose tissue, and the blue color is subcutaneous adipose tissue. The body composition can quantify in two ways. The first way is the tissue index, based on the tissue's area adjusted on the patient's height. For example, like the skeletal muscle index. The other way is focus on the tissue radiation attenuation. Look at the original CT scan based on the CT value, also Hounsfield unit. For example, like the skeletal muscle radiation attenuation, so-called myostosis or non-myostosis. Based on the background, we hypothesize the clinical models based on body composition parameters can be used to identify cancer patients who may benefit from immunotherapy. And we're also wondering how about the changes in body composition during immunotherapy interact with the tumor immune microenvironment. Let's move to the study 1. We involved 120 advanced gastric cancer who received the chemotherapy plus a PD-1. Follow the criteria. The most important thing is the patient should have the high quality number 3 CT scan before treatment. And we give the 3 or 4 cycles immunotherapy. After that, the patients will receive the surgery. We focus on the parameter like the body composition, skeletal muscle index, and the radiation attenuation and the adipose tissue parameters. And we also focus on the systemic inflammation like the neutrophil lymphocyte ratio and the body weight loss. We have 2 research questions. The first one is the short-term goal, the tumor regression grade. We follow the guideline to give the grade. And the second research question is the long-term goal. We focus on the survival, include the OS and the PFS. First, we use a regression to identify all of the clinical parameters and the good responder, this group, and the poor responder, this group. In the end, we only found 3 parameters: the skeletal muscle radiation attenuation and the NLR and the weight loss were significant with the result. We want to know more detail, so we use Lisa Martin's cutoff, depends on the female and the male, but adjusted by the Chinese population BMI. We can divide the high and the low skeletal muscle radiation attenuation, and we use the ROC curves, the Yoden index, to divide high and the low NLR, as well as we use a 5% to divide the weight loss. In the end, we still can see the skeletal muscle radiation attenuation, NLR, and the weight loss is associated with TRG. Let's move to the survival. First, we're still wondering how about the body composition. So via the survival curves, we can see if the patient with a higher skeletal muscle, these curves, they have a better survival, both in PFS and the OS. We're also wondering how about the NLR. So we can see if the patient have a lower systemic inflammation, they have a better OS. As well as the weight loss, we can really see they have the attendance. If the patient lose less body weight, they may have the better survival, but no significance. So that may suggest if we consider the cancer cachexia should be beyond the weight loss. But we know if we see the patients, if cachexia or no cachexia, maybe combine the different phenotype. So I want to combine the different parameters. We follow this group, a better body composition, like the high skeletal muscle radiation with or without the other risk. So we can see both in PFS and OS, if the patient have a good body quality, skeletal muscle radiation attenuation, they always have a better survival than if the patient have a lower skeletal muscle radiation attenuation. If we build predict clinical models, we always think if either any confounder affects the result. So I would like to use the Corsica regression analysis. In the multivariable analysis, we found the skeletal muscle radiation attenuation is a independent factor for the patient's survival. In conclusion, the first one is the skeletal muscle radiation attenuation is associated with physiological response, so-called tumor regression rate, and the survival of gastric cancer patients who received with chemotherapy plus PD-1. The clinical model consisting of baseline skeletal muscle radiation attenuation, NLR, and weight loss can be used to identify gastric cancer patients who can benefit from the immunotherapy. And in the future, we would like to involve the basic science to explain the phenomenon. Just now we talk about the baseline body composition, but I'm still curious, how about the body composition change during the immunotherapy? I would like to use the case of the cancer patients. In visual, we can see the before and after the immunotherapy, their body composition can be changed. So let's move to the study 2. This time we involved 81 advanced lung cancer patients who achieved the immunotherapy and 190 patients without immunotherapy. The same criteria, but the only different thing is we have the follow-up CT scan when patients finish the treatment. This time we focus on the parameter body composition And we also have the data of circulating T cell from the blood. And we also have the tumor infiltrating lymphocytes from the tumor tissue. We have two research questions, include overall survival, and we also want to understand this interaction between body composition and immuno-microenvironment. Still same, we can see the skeletal muscle index loss is associated with poor survival in whole cohort and immunotherapy cohort and non-immuno cohort. The similar reason we want to know is the body composition is independently affect the patient's survival. We use a cost regression analysis for all of the cohort. But the very interesting thing is we found the skeletal muscle loss is the only one factor for the poor survival in immunotherapy cohort. So that's raises a big interest to know how about the body composition, especially the skeletal muscle and the immuno-microenvironment. As I mentioned just now, we have the 48 patients who have the circulating T cells. So we use a logistic regression to analyze both the baseline and the change after the treatment. We can see the similar result. The CD3, if the patient have a higher CD3, they have a poor baseline skeletal muscle index, and they more easily to lose a skeletal muscle index. But for the CD8 play a predictor factor for the baseline skeletal muscle index and the change of the skeletal muscle, I mean the loss. So it proves us a clue from the skeletal muscle to the tumor. As I mentioned, we have the 24 patients' tumor tissue, use the same method to divide the skeletal muscle stable and the skeletal muscle index loss. We can clearly to see in this figure, if the patient have a higher CD8, they are more easily to keep the skeletal muscle index. Although in this figure, there is no difference for each other, but we can see a tendency. If the patient have a higher helper T cell, maybe they have a stable of the skeletal muscle index. In conclusion, the skeletal muscle loss is an independent predictor of poor prognosis in patients treated with immunotherapy. Higher relative abundance of peripheral CD3 cells and lower relative abundance of CD8 cells is associated with skeletal muscle index loss. And the higher proportion of CD8 tumor infiltrating lymphocytes is associated with skeletal muscle index stable during treatment of advanced lung cancer patients who receive immunotherapy. Before I thank for our group, I want to give a short summary. In the future, we would like to involve some muscle biopsy and the blood circulating T cells and the tumor tissues combined with radiology based on the AI technique for help us deeply understand the cancer cachexia. Thank you.
Thank you, Jan. So if you have questions, please write in the Q&A. So I didn't notice, Jan, in your two cohorts, did you have both male and females?
Yes.
Okay, and did you notice any sex-dependent behavior or?
Yeah, actually for both cost regression analysis and the logistic regression analysis, we always considers a female and a male as a control confounder, but there is no difference for the, there is no significance for the result. I just put the significance result into the slide.
Okay, so you analyzed your CT scan images. So did you also try to analyze the fat or the fat infiltrating the muscle, so the myosteatosis? Did you consider also these parameters?
Yes, actually, we analyze all of the parameters, including the visceral fat, no matter index, and the radiation attenuation, and the subcutaneous adipose tissue index, and the radiation attenuation. But after the multivariable analysis, there is no significance for the fat, so only the significance in the muscle.
Okay. So Joanna Karizinska is asking, how do you define the skeletal muscle index loss? Is there a threshold value? And the same question, and are women and men affected differently?
Yeah. I think in the future, we want to improve our study because you can realize our sample size is not too big. So normally, in every cohort, the female is less and the male is bigger. If we have more data, I think I will set the sub-cohort, really focus on the female and the male to see how about the immunotherapy affects the body composition, or the body composition can affect the immunotherapy response.
Okay, so we have a related question from Laurie Westerlund. So does immunotherapy help patients gain muscle back after having severe cachexia?
Actually, it's really difficult to understand, to answer. In our cohort, if the patient receives immunotherapy, they have a better survival. But for gain muscle, I'm not really sure. Right now, I only can see in our studies the cytotoxicity T cells, the CD8 can help the people gain or stable the muscle. But for the other lymphocytes, I'm not really sure. There is a review I can share with my screen.
Yes.
If you are interested, you can look at this review. For the CD4, the helper T cell, sometimes it really help for the cachexia, but sometimes it can anti-cancer cachexia. So maybe if somebody interested in this field, you can continue the research. But right now, I can't really answer the question.
Thank you. So
Yeah.
there are a couple of more questions. So Maria Stilander, so thank you for the great talk, Jan. Have you screened for wall tumor secreted cytokines changes after immunotherapy? Have you screened for whole tumor secreted cytokines changes after immunotherapy?
No, I would like to involve, but we only have the data before the treatment. I'm really curious how about the cytokines after immunotherapy. So you give me a very good suggestion for the future.
Cool. So the last question from Sarah Judge. So you mentioned that higher CD8 positive tumor infiltrating lymphocytes was associated with stable skeletal muscle index in advanced lung cancer patients receiving immunotherapy. Was this finding also associated with a reduction in tumor-associated macrophages?
It's a pity because we have a lot of missing data of that, but I would like to involve. As I showed the review just now, it also plays a role in cancer cachexia anti or predict. So I would like to do it in the future.
Okay.
Thank you.
Thank you, Jan. So the time is over. I really would like to thank the three speakers. It was a fantastic session, and thank you all again for participating. And just a quick reminding from everyone. So if you are a member of the Cancer Cachexia Society, you have the opportunity to post the job advertisement in the Cancer Cachexia Society website. And I invite you all to check for the job ads in our website. So thank you again, all the speakers, and see you in the next seminar that will be held in October 9. Thank you again, everyone.
Thank you.
Thank you. Bye.